So our next speaker is Alexander from the MRC, who's going to take us through the work towards generating or driving scattering factors for cryo.
Okay, great. So, yeah, today I'm going to talk about some of our work addressing some of the shortcomings of the scattering models that we currently use in order to model electron microscopy data. So, to briefly outline the talk, I'll introduce the problem first. Then I'll talk about an algorithm that we've developed in order to infer scattering amplitudes directly from cryo-EM maps. And then finally, I'll present some results on applying that algorithm to real EM data. Okay, so I guess a little bit of a change of focus compared to some of the other talks. I'm going to skip right to the end of a successful cryo-EM experiment. And the product of that is a map that represents the electrostatic potential in the molecule that you're studying. And that's useful to us as structural biologists because the electrostatic potential contains a lot of information about the molecule that we're interested in. So first of all, it's influenced by the positions of the atoms, the atomic coordinates. And we can use that to refine an atomic model and then deposit it in the PDB. But the electrostatic potential is also strongly influenced by these other factors: charge on atoms and ions, and delocalization of electrons due to bonding. And we'd like to be able to extract that information directly from EM data. We'd like to be able to account for those effects in electron microscopy, electron scattering experiments. At the moment, we can't do that. So why is scattering at all relevant? Cryo-EM is in essence a scattering experiment. So I have here a very simplified diagram that shows an electron microscope. You have your specimen on the top that gets irradiated by an electron beam. And the wave that forms at the exit face of the specimen is formed of an unscattered contribution and a scattered wave that's formed by interaction of the incident wave with the sample. The exit wave is then focused down by the objective lens in order to form an image. And we observe in the image the intensity of the exit wave plus the effects of of various aberrations, which we assume that we've corrected for by the time we get to the final map. So in order to understand image formation, in order to understand what we see in the micrographs that we get out of an EM experiment, we need a good model of this exit wave formation. And the model we use has several components. The first one is this weak phase approximation. So we introduce this quantity, the scattering amplitude, in order to describe the interaction of the effect of the specimen on the incident wave. And under the weak phase approximation, the scattering amplitude is simply the Fourier transform of the electrostatic potential of the specimen. And you can derive this by assuming that the wave undergoes a single scattering event as it passes through your sample. And for those of you that are interested, this is called the first Born approximation in physics. Microscopists tend to call it the weak phase approximation. Weak phase approximation is useful because it relates the scattering and the electrostatic potential by a simple linear transform, the Fourier transform. And it means that we can choose to model either one of these quantities and interconvert simply between them. I haven't spoken yet about the form of any of these functions, the electrostatic potential or the scattering. And at the moment, the model that we use for scattering is this independent atom model. So we assume that the scattering from a molecule can be expressed as a sum of atomic contributions that we call atomic scattering factors. And that these atomic contributions are radially symmetric around the center of each atom. And we ignore any effects due to the interaction of atoms within the molecular context. So bonding, ionization, screening by solvent. And what this means is that every atom in your molecule scatters in the same way as under this model, scatters in the same way as a free atom floating around in a vacuum. And so we can pre-calculate these scattering factors for every element and tabulate them. And then when doing calculations, we can just refer to these tabulated scattering factors. So I'll refer to this current model as the tabulated scattering factors in the talk. Now, our aim is to try to address some of the shortcomings of this model while keeping its computational tractability and develop this kind of semi-independent atom model in which the molecular scattering is still a sum of atomic contributions and atoms are still radially symmetric and we still ignore solvent effects, but we would like to account for the main source of error in this model, which is bonding and ionization. We'd like to account for those effects. So a more concrete example that I've given here, I've plotted the scattering factors for two chemical species, a free oxygen atom and an oxygen anion. And on the x-axis is frequency. So you can see that at high frequency, above about 2 angstroms, the two species scatter almost in the same way. On the other hand, at low frequencies below 2 angstroms, and this corresponds to the resolution range that we see in an EM experiment, they scatter really differently. And what this suggests is that if you have negatively charged functional groups in your protein, they should scatter really differently to atoms of the same element that aren't negatively charged. And we should be able to observe those effects in maps. So that's one of the research questions that we're seeking to address. And the way we want to address this is using a data-driven model. We know that data-driven kind of machine learning approaches have been really successful in various fields. And also in EM, we have this exponential growth in the number of EM structures that has been deposited to the PDB in the last decade or so. And the question is, can we use this data in order to learn the scattering factors directly from the data, and in this way bypass expensive computational calculations that would otherwise be required? And we've developed an algorithm that does this. The way we want to approach this is using Bayesian machine learning. So a quick recap of how to define a Bayesian model. In a Bayesian model, we have to define two probability distributions. The observation model, which in this case describes the probability of observing a particular cryo-EM map given a set of scattering factors. The prior distribution, which encodes our beliefs about what scattering factors should look like, independent of carrying out any observations. And then we use these two distributions in order to derive the posterior distribution, which gives us the probability of a particular set of scattering factors given a set of cryo-EM maps. And we use this posterior during inference, so we can find the set of scattering factors that then maximizes that probability. So I'll go through the observation model and the prior in turn. Our observation model is based on the weak phase approximation and on the independent atom model. So the Fourier transform of the cryo-EM map, that's the part that's due to the weak phase approximation, we say is a sum of atomic contributions. But now, instead of having one scattering factor per element, we're going to have one scattering factor per atom type. I'll talk a bit later about what I mean by an atom type. And in order to make this problem easier, we assume we know some properties of the molecule in advance. So we assume that we know the locations of the atoms. And we also assume that we can parameterize the uncertainty in atomic positions using B factors. And we can get those parameters using a model refinement program, such as RefMac, for example. And we want to model real data that has noise, so we need a noise model. Our noise model is independent Gaussian noise in Fourier space, and that's fairly standard for cryo-EM data processing. So a word on these atom types. As I've mentioned, at the moment we have one scattering factor per element that's pre-calculated and tabulated, and we want to generalize that approach. And now we're going to say that atoms of the same element that have the same set of bonding neighbors share a scattering factor. And this is a way of accounting for the effects of the environment, of the chemical environment, the effects of bonding, basically. As a side effect, all proteins are polypeptides, so common chemical environments will be present in all proteins, which means that we can use many maps simultaneously in order to infer scattering factors, and in this way, hopefully, increase their accuracy. Here I have an example of how atom typing works. So let's say you have this backbone carbon atom. So our algorithm would assign the atom type carbon bonded to carbon, nitrogen, and oxygen to this atom. And it would be grouped with other atoms that have the same set of neighbors. Okay, in a typical protein, there are about 40 atom types. And I've given the most common ones in a molecule of catalase here. So you can see that the most common atom types basically correspond to functional groups found on the backbone. And they contain, the most common atom types also contain a very large number of representatives. So that's the observation model. To summarize quickly, the way we generate observations, you can see that. So is we have these atomic contributions, and we sum them together in the presence of noise in order to get this noisy summation. And our problem is to go from the observations in the bottom row and to try to decompose it into atomic contributions. Now, that problem by itself is ill-posed. So we need to define a prior distribution over these atomic contributions in order to solve it. We use a class of prior called a Gaussian process prior, and the characteristics that this prior encodes are ones that we can observe in the tabulated scattering factors that I've plotted on the right here. So we want our scattering factors to be smooth functions of frequency. We want them to decay to 0 as the frequency goes to infinity. And we also want them to change slowly at low frequency. And this last requirement is something that we observe in the tabulated scattering factors. And we would expect it to be true for all neutral atoms. Okay, so we've implemented this algorithm, and we've applied it to some data. Because this is a regression problem, we've split our data into a test set and some training sets. So our training sets are, the first one is small. It consists of 3 catalase structures solved by our colleagues at the LMB. I'll talk about them on the next slide. And the second data set is much larger. It contains these 52. It contains these 52 maps from the MDB that have resolution better than 2 angstroms. And the test set for all of the results I'm going to show is a set of 12 maps from the MDB also with a resolution better than 2 angstroms, but importantly, they're different to the 52 maps that we used for training. Okay, so this work on the catalase data sets was really driven by Hugh Wilson and Richard Henderson in the lab. They've solved to high resolution, so sub 2 angstroms in all cases. The structures of catalases from 3 species, 2 bacterial species and human erythrocyte. And they've used hexafoil grids in order to reduce artifacts due to specimen motion. The metric I'm going to use for these results is the map model FSC. So the way this works is we take a cryo-EM map from the test set. And on the other hand, we take an atomic model that's been refined into this cryo-EM map. And we take a set of scattering factors that's been calculated using one of the training sets. And we use that to calculate a map, a simulated map, if you like. And then we calculate the FSC between our observed map and the calculated map. And we can do this using the tabulated scattering factors and the new scattering factors. And hopefully we will see some increase in the FSC. So positive values in this case are good. It means that we get improved agreement with the data. So I've done this for the scattering factors that have been determined from our 3 catalases using, and I've plotted this FSC change for all of the maps in our test set. And what we can see is that we do get some increase in the FSC in a region between 15 and around 3 angstroms, with a peak improvement of around 11 percentage points. We can do the same thing using scattering factors determined from our larger training set from the MDB. And the shape of the curve looks very similar, but the peak improvement is slightly larger. And that's encouraging. It means that if you put more data in, you get scattering factors that perform better on the test set. What do these scattering factors look like? These I've plotted here scattering factors for 3 elements determined using these catalases. And each line in this case represents one scattering factor for an atom type. So you can see that at a high frequency, they agree with one another. All of the scattering factors are reasonably similar, and that's expected. That's in line with what we expect. At low resolution, there is, however, quite a lot of spread. In particular, what is interesting is that there are some atom types that appear to be outliers with respect to other atoms of the same element. So, for example, this carboxyl carbon atom or this amide nitrogen. And at the moment, we can only speculate about why that is. It could be because of radiation damage. So, for example, these carboxyl groups are known to undergo radiation damage. Could be due to some charge effects or some other artifacts. So we're in the process of investigating this. And the final piece of data I want to show is something that's, in a sense, orthogonal to cryo-EM. And it relates to predicting a quantity of materials called the magnetic susceptibility, which describes the ability of a bulk material to magnetize in an external field. And it turns out that the magnetic susceptibility is, to a good approximation, proportional to the sum of the scattering factors at 0 frequency. So the way you would calculate this is you would look at your molecule, you would look at every atom in the molecule, right? For that atom, you would look up the 0 frequency, the scattering amplitude at 0 frequency, so this kind of values here, and then you would sum them together. And this value has been measured empirically for hundreds of small organic compounds. So what we can do is we can take the empirical value on the one hand, on the other hand, we can try to predict that value, and then we can compare them. So I've done this here using scattering factors from the tabulated scattering factors, which are in orange, and the scattering factors that have been fitted to this catalase data set. And in this plot, each point is a single small organic compound. It's the magnetic susceptibility for a small organic compound. And ideal predictions would align along this line, along this black dotted line. So you can see that if you use tabulated scattering factors, you get some kind of systematic over-prediction of the susceptibility. On the other hand, if we use scattering factors determined from these catalase maps, we appear to be able to remove that systematic error, even though we get more variance in the predictions. So that's quite encouraging. So I went ahead and did the same thing for scattering factors from the EMDB maps. And unfortunately, it doesn't seem to work. So you get these very high variance predictions that are also different from the measured values. And we think that the reason for that is something to do with data quality. So basically, these catalase molecules are very rigid, and they've been also modeled. The models have been built very carefully into them. And so we can expect that the signal is pretty uniform across the map. Whereas in the EMDB, we have all sorts of cases of more challenging proteins that might have flexible regions, or they might have regions that haven't been modeled very well. And so that will, of course, impact the predictions. And so this is an argument for using really carefully modeled, really high quality data for these calculations. Okay, so to summarize, we've proposed a method to learn scattering factors from cryo-EM data. And the scattering factors produced by this method show improved agreement with observations compared to scattering factors that we use at the moment. And finally, we get reasonable performance using very small training set. And that's encouraging because it means that in future, we could apply this algorithm in order to calculate scattering factors from just a single cryo-EM map, for example, and use that to try to infer some effects of interesting atoms or functional groups. Future directions. On the data analysis side, we would like to be able to link these scattering factors to actual chemical properties or biochemical properties. For example, charge or protonation states. And the other main issue is radiation damage, which I've only touched on briefly in this talk, but it will definitely affect the results, so it needs to be accounted for. And we're working on new atomic model refinement algorithms in order to address that. And we're also working with the zero dose program from Jude Short, from Chris Russo's group, in order to produce damage corrected maps. There are also some theoretical questions. The first one is hydrogen atoms, which we would expect to be heavily polarized by the atoms that they're bonded to. And so the suggestion is that we would need a different parameterization for hydrogen atoms compared to other atoms. And in order to develop that parameterization, comparison with theoretical quantum chemical calculations on small molecules will be really key. So I want to thank everyone who was involved in this project. First of all, my supervisor, Gary, as well as colleagues in the group. And on the experimental side, I think I want to point out particularly Hugh and Richard, as well as Jade Lee, who did the model building for these catalases, and Daria, who purified the proteins. I also want to thank Shaw's and Ben for productive discussions and the fantastic facilities that we have at the LMB, without whom this work wouldn't have been possible. Thank you.
Thank you very much. Are there questions from the audience? If not, I'm going to take the first question. Again. So obviously, for me, deriving charge information for sites, for ligands, and all the rest of it is really exciting. So, what's the next step, do you think, for us to be able to extract and infer where the charge is in, for example, a local environment in a protein?
Yeah, so that's an interesting question. Basically, in order to partial charges in molecules are not actually observable, right? There's something we have to derive. We have to define them and derive them somehow from the potential. And in order to do that, you need information about how the potential behaves very far from the molecule, which is equivalent to modeling these low resolution components and scattering factors. Okay. So one direction is to try to improve that part, which is probably the most challenging region to model. The other possibility would be to basically convert these scattering factors are in Fourier space. So we would just do an inverse Fourier transform, convert them into real space, and then try to fit some kind of charge to that.
Yeah, that makes sense. I have a question on Slack from Randy. So it's asking if you could prune your larger training set to remove atoms from regions that have lower confidence in the fit or higher B factors.
So I didn't mention this, but we are doing this already. So I'm filtering atoms with B factors that have higher than some threshold.
Right, yeah. So they're already not included.
Yeah.
Is there a question? Oh, yeah, thank you.
Thanks for the nice talk. Regarding this problem with having this large data set, you're showing that it's actually performing better, but then you're showing that it's actually, so how do you explain that?
Right, so these two metrics are sensitive to different things. The FSC is not sensitive to any isotropic scaling of the data, so it's a correlation coefficient, right? You can multiply one of the sets of values by some value and it won't affect it. On the other hand, these magnetic susceptibility predictions are actually sensitive to the value, the actual value. So if you multiply your scattering factors by 10, you'll get multiplication by 10 of the prediction. So they're measuring different things basically.
But it's still problematic because you know that it's not accurate, and then yet you're suggesting it for improvement. But maybe cleaning your data would be.
I think that's probably the way forward.
A final question there, I think.
Hi, thanks for the talk. Very nice, different perspective look at data. So what is your computational cost to look at this? Like if we want to look at lots of data from EMDB, want to use that to validate the model. Yeah, how long does it take for you to those 52 or 12, all of that?
So I haven't actually benchmarked this. Running on a GPU on an A100, it doesn't take very long. So a single map takes less than 1 minute maybe to do the inference. The main bottleneck is it's very memory intensive. That's the main problem.
Thanks. One final question up there, and then I think. Sorry, your microphone's off.
Okay.
Thank you.
Very nice talk. Thank you. You touched briefly beam damage, but you didn't put in more. If you have your movie and you can do the different frame averages, somehow can you monitor what goes down first just by looking at your charged sites?
Yes, so we're working on this at the moment, actually. The idea is basically to do, you can generate one map from every frame of your movie, and then you can simultaneously refine a model of the damage and the scattering factors using that.
I think this would be very important to compare then images taken at helium temperature versus the normal nitrogen.
Yes, so when that is ready, then we can start using it.
quite helium everywhere yet. Brilliant. Thank you very much, Alexander.
Okay.
Thank you.